In this lecture, we're going to speak about the lymphatic system and immunity. Lymphatic system consists of lymph vessels. It consists of lymph, the fluid that flows through the lymphatic vessels. It consists of lymph organ, and it consists of lymphatic tissue and cells. So what is the function of lymphatic system? The primary function of lymphatic system is to maintain and distribute lymphocytes. And lymphocytes are the cells that basically protects and defend your body. In addition to the lymphocytes, you have other cells like natural killer cells, macrophages, and all other white blood cells that protect and defend the body. In addition to this one, the lymphatic system is also responsible for maintaining fluid balance and absorbing fat. So what it does is it absorbs basically vitamin soluble fats and fat soluble vitamins and fats from the digestive system and subsequently transport those substances to the circulation. And it also returns excess interstitial fluid back into the circulation. So let's look at the lymphatic vessels. So lymphatic vessels are basically a network of tubes located throughout the body. They transport lymph away from the tissue, and they transport lymph toward the lymph nodes, and then from lymph node they pass through the big ducts and then drain into or empty into the subclavian vein and into the circulation. So as you see from this picture over here, so this is the lymphatic capillary. It will basically collect excess interstitial fluid. So excess interstitial fluid will enter the lymphatic capillaries. Many lymphatic capillaries will collect, and they will become collecting lymphatic vessels. So all that you see over here, those will be all collecting lymphatic vessels. So many capillaries will drain into lymphatic vessel or empty into lymphatic vessels. Lymphatic vessels will deliver or pass through many lymphatic nodes. So this is the lymphatic node over here. So you see this dots over here. Those dots represents lymphatic nodes. So you see a bunch of lymphatic nodes here. So the lymph that goes here will pass through those nodes, go further, and then again you see nodes here. It will pass through these nodes and eventually empty into the lymphatic duct. So the lymphatic vessels that deliver the lymph into the lymphatic nodes are called afferent lymphatic duct. So this is how they enter. So this is all basically entering. So this is the afferent lymphatic duct. Many afferent lymphatic ducts will enter the lymphatic node. We're going to discuss the function of lymphatic node later, but this is mainly monitoring the flow of this lymph, and if there's any sign of infection, the lymphocytes will be activated. The efferent artery or the efferent lymphatic vessels take the lymph away. So the efferent takes it away, the afferent brings the lymph in. The efferent lymphatic vessels basically will empty into a very large duct, either right lymphatic duct or thoracic duct. And thoracic duct and lymphatic duct will enter and empty into right and left subclavian vein. So let's look at the lymphatic capillary. What are lymphatic capillary? They are a very fine network of tubes that's found throughout the body. They serve as a drainage for excess interstitial fluid. There are a few regions in the body that don't have lymphatic capillaries. That would be your dermis of the skin. So the dermis of the skin doesn't have lymphatic capillaries. Mucosal membrane, bone marrow, and central nervous system also do not have lymphatic capillaries. There are some regions that have more packed, they're more packed with the lymphatic capillaries, and those would be the dermis, the lungs, the gastrointestinal and genitourinary systems. Now, let's look at the lymphatic capillary in more detail. So lymphatic capillaries begin as a blind end. If you compare the blood capillaries, so you see the blood flows. Let me change the color so you could see it into blue. So the blood flows. This is the arteriole. So the blood comes from the arteriole. As soon as that arteriole branches into smaller vessels, now we call them capillaries. And then from the capillaries, as they join back together, as those vessels join back together, now we call them venule. So it's kind of like a continuous tube. Doesn't have beginning, doesn't have an end. It's just continuous. If you look at the lymphatic capillaries, they're beginning as a blind or close-end ending, and they're more permeable compared to the blood capillaries. In this picture, what you see over here is that you see this one represents the cells over here, and everything else that you see over here represents the basically lymphatic. So everything that you see over here, that basically represents the interstitial fluid, and so the blood or some fluid from the capillaries come out, and that excess fluid ends up in the interstitial fluid, and that builds up pressure, and that pressure pushes the fluid into the lymphatic capillaries, and the lymphatic capillaries will go to the lymphatic collecting ducts and eventually deliver it back to the circulation. So they're basically, when you look at the lymphatic capillaries, they are formed by endothelial cells, and that endothelial cells are like a button-like junction between them. So there's like a button-like junction in between those endothelial cells, and those button-like junctions allow the fluid to pass through them when the interstitial fluid pressure is sufficiently high. And this picture over here again shows you, so right over here you see the cells over here, so that's representing the cell, and this is the nucleus. So obviously between the cells, let me change the color to a different, so right over here between the cells you see a lot of interstitial fluid. So all this excess interstitial fluid, as the pressure builds up, they will just flow into the lymphatic capillaries. So lymphatic capillary begins as a blind pocket, and then it has this switch-like endothelials that the fluid goes in, and the fluid is transported to the collecting ducts. And the other thing that you're going to notice about the lymphatic capillaries, that they have valves in the lymphatic vessels. So you see they have those valves. And if you look at this valve, the valve is open and the fluid moves in. And this valve is closed, so it basically prevents the fluid going back this way. So the fluid cannot go backward, and the fluid could only move forward. So doesn't allow the backflow of the fluid. And those junctions are basically protein filaments, and they're collagen fibers, and they serve as a valve. So you see those valves are basically collagen fibers, and they prevent back leaking. The other function of the capillaries, we said they absorb and deliver fat and fat-soluble vitamins. So let's say that this is the intestinal lumen. So right over here you have glucose, you have vitamins and everything. You have monosaccharides, amino acid, you have fatty acids, glycerol. So everything has to be transported. Everything has to cross from the microvilli of the intestine, enter the cell, and then from the cell enter the circulation. So let's look at a close-up picture over here. So this is what happens. So when you have the amino acids will cross, go to the cell, and then from the cell they will enter the circulation. When you have monosaccharides, they will also go to the cell, and then from the cell they will cross into the circulation. But fatty acids and glycerols, they will go to the cell. They will be packed with the lipid transporting protein, lipoproteins, and then they will cross into the lymphatic capillaries because they're too big to be to cross into the blood capillaries, so they cross to the lymphatic capillaries. Remember, we said lymphatic capillaries are more permeable. It could take more larger molecules. And this portion and this lymphatic capillaries are called lacteals. Now, from the lymphatic capillary, the lymph is going to move and be delivered to the lymphatic collecting lymphatic ducts. So those are all collecting lymphatic ducts that you see all throughout the body, and those are all collecting lymphatic ducts. So collecting lymphatic ducts, as they're going, they will pass through the lymphatic nodes. So you see a lot of different lymphatic node collections: cervical lymphatic nodes, upper arm, you have the intestinal, you have abdominal. So you have a bunch of those lymphatic nodes throughout the body that the lymph passes through. So lymphatic nodes will deliver it to the either right, all the collecting lymphatic ducts will either go to the thoracic duct or to the right lymphatic duct. And what is going to happen is the thoracic duct is this guy right over here. This is the thoracic duct. Lymphatic duct is right this one over here. This is lymphatic duct. So what is going to happen? Thoracic duct is going to collect the lymph from this portion of the body. So thoracic duct is basically collecting lymph from lower limbs, from the diaphragm, from the left arm, left side of the head, neck, the thorax, and everything empties into the left subclavian vein right over here. And the right lymphatic duct basically collects the lymph from this half of the body. So from the right hand and a portion of the right head, the lymph is collected by the lymphatic duct. So we could say that 75% of the lymph is basically collected by the thoracic duct. So almost like 3/4 or 3/4 of the lymph is drained by the lymphatic duct, and 1/4 is delivered by the right lymphatic duct and delivered and emptied into subclavian vein. We're going to look at the subclavian vein in a second. Now, the lymphatic thoracic duct over here, the thoracic duct begins with a pocket. It has like a big pocket in the abdominal. It looks like a sac, and this sac in the abdominal region, that's called cisternae chili. So cisternae chili is this sac which is the beginning of the thoracic duct. So you see this is the thoracic duct going up here. So let's look at it at the picture. So you see right over here, that's the cisternae chili right over here, and this is the thoracic duct. And you see how thoracic duct goes and empties into the subclavian vein. And now we're going to look at that subclavian vein. So you see this one is over here and this one over here. So you have the right and left subclavian vein. This guy over here, this tube that you see over here, that's the thoracic lymphatic duct. And this is the right lymphatic duct. So the right lymphatic duct, we said, takes approximately 25% of the lymph and brings it to the circulation. And the thoracic duct, we said, brings approximately 75%. So you see thoracic duct begins with a sac and then it moves up. It moves up and then it goes and empties into the left subclavian vein, and right lymphatic duct empties into the right subclavian vein. So what is lymph now? So we discussed the lymphatic vessels. Lymph is basically a fluid that's very similar in composition to the blood plasma. It's basically delivered from blood plasma as a fluid that passes from the capillary walls in the arterial end, and it ends up in the interstitial fluid. And as it accumulates in the interstitial fluid, partial pressure in the interstitial fluid increases, and that partial pressure forces the fluid to move to lymphatic capillaries. And as soon as that fluid enters into the lymphatic capillaries, we now call them lymph. And that lymph will go to the collecting ducts and eventually to the large thoracic and right lymphatic ducts and empty back into the circulation. So it's kind of like circulating, recirculating or returning the fluid back into the blood. Now we're going to look at the organs of lymphatic system. Lymphatic organs are characterized by cluster of lymphocytes and other cells such as macrophages and M cells, dendritic cells, and others. So it will have cluster of those cells. The lymphocytes, not only lymphocytes, all the cells are originating in red bone marrow. So regardless of what the question asks you, if they ask you where is macrophages produced, they're produced in the red bone marrow. Where are T cells produced? They are produced as lymphocytes in the red bone marrow, but they differentiate and mature in the thymus. So all the cells are produced in the red bone marrow, but then they're carried or migrate from the red bone marrow to the lymphoid organ, where they're exposed to the microorganism and they mature and proliferate. So what are the primary lymphoid organs? So the primary lymphoid organs are two organs, the thymus and the red bone marrow. So red bone marrow and thymus are the two primary lymphoid organs. What are the secondary lymphoid organs? Secondary lymphoid organs are the spleen, the lymphoid nodes located all throughout that you see, Peyers patches that's located in the digestive system right over here along the intestine, mucosal associated lymphoid tissue that we're going to discuss. This is also located throughout the digestive and other mucosal membranes, and tonsils. So now let's look at the thymus. What is thymus? Thymus is basically an organ that's located behind the sternum in the chest. It reaches its maximum size just before puberty, and it's basically consists of two lobes. It's lobed organ. It consists of lobes, and each lobe is basically consists of cortex and medulla. The lymphocytes in the cortex are divided, and when they're mature, they migrate to the medulla, and after about 3 weeks, they leave the thymus and they enter the circulation. So what is the function of the lymphatic system? Function of lymphatic system, the primary responsibility is production and maturation of immune cells. More specifically, maturation of T lymphocytes, T cells. So lymphocytes are produced in bone marrow, and lymphocytes, so what you have is bone marrow right over here. Let's say that this is the bone marrow inside. Inside you produce lymphocytes. Some lymphocytes stay in the bone marrow. Some lymphocytes leave the bone marrow and will enter the circulation. Circulation will transport that lymphocytes to the thymus. In the thymus, they will mature into T cells. The ones that stay in the bone marrow, they will become either B cells or natural killer cells. So the NK cell and B cells, bone marrow cells, lymphocytes are staying in the bone marrow. The T cells or thymus cells or thymus lymphocytes migrate, leave the bone marrow and goes to the thymus and mature there. The other function of the thymus is that they produce hormones such as thymosins, thymopoietin, and thymolene. And in addition to that one, they also stimulate, we said, division and differentiation of T cells. In addition to that, they also produce signaling molecules such as interleukins and interferons. As humans age, the thymus basically shrinks, the size decreases, and that decrease in thymus size may basically make the person more susceptible for diseases. Red bone marrow is involved in homeostasis. We said all the bone cells are produced, all the blood cells are produced in the bone marrow So if you look at this one over here, so this is the production of the blood cells. This more specifically, this one shows you that megakaryocyte is basically exploding and creating those tiny platelet cells. But the same way other cells will be produced. So all the cells basically produce in the bone marrow, and they migrate through these tiny gaps into the sinuses, and then from the sinuses they will enter to the blood circulation. So what is bone marrow? Basically it's a soft, very soft, loosely organized, highly vascular, has a lot of blood vessels, tissue. And what happens in the bone marrow? In the bone marrow, the blood cells mature. As they mature, they will push their way through the reticular and endothelial cells, and they will enter the sinus and flow away into the circulation. Spleen. What is spleen? It is basically located posterior within the peritoneal cavity in the upper left quadrant of the abdomen. So if you look at it like this, it's located right over here. If you divide the abdominal cavity into four, it's located in the upper left hand side of it. The spleen contains two major units, and those two major units it consists of basically two major parts, white and red pulp. White pulp is basically where you find a lot of like white B cells and T cells. So B and T cells are found mainly in the white pulp. Red pulp is where you mainly find macrophages and monocytes. There is also red blood cells located there. So what is the function of spleen? The function of the spleen is basically filtration of the blood cells, filtration of the red blood cells and platelets. This filtration occurs in the red bulb. So the red bulb, in the red blood bulb, what happens is the red blood cells pass through the spleen, and the spleen basically traps older or damaged red blood cells and deformed red blood cells, and those that are young and cannot be are not damaged, they could just pass through. So the red blood cells that are trapped into the spleen will be phagocytized by macrophages. So once macrophages phagocytize them, they will break down those red blood cells, and I will basically separate the iron from this red blood cells and amino acids from the red blood cells. And when the red blood cells, macrophages basically take this iron from the red blood cells, they will do one of the two things. Either they will store it within the cytoplasm, or so the macrophage will store it within the cytoplasm, or they will export that to other parts back, for instance, like to the red bone marrow, so they could make more red blood cells. In order to make red blood cells, you need iron. So they will export that with the help of transport protein that's called ferritin. In addition to that one, the function of the spleen is basically prevention of infection. So infection is prevention occurs by two major mechanisms. One is that phagocytes basically monitor as the lymph passes through the spleen, and monitor for any sign of antigens or pathogens and anything, and they just engulf it and destroy it. The other way of looking at it, they produce antibodies. So basically, if macrophage encounters a pathogen, it will destroy it, and at the same time, it will present the antigen to the T helper cell. And T helper cell will stimulate the B cells, and B cells will proliferate and then become plasma B cells, and plasma B cells will produce antibodies. In addition to that, spleen could also serve as a storage for red blood cells and platelets. And also removal of obviously abnormal red blood cells. We said whenever there is like abnormal red blood cells, macrophages will just engulf them and destroy them. Lymph nodes. What is the shape? So lymph nodes are basically like a kidney, small kidney shape or oval shape structure. They're basically, each lymph node is surrounded by a fibrous connective tissue. So this green one that you see over here, that's the fibrous connective tissue. So you see this fibrous connective tissue? All. Now you see some of the fibrous connective tissue extends into inside. So this fibrous connective tissue that surrounds the lymph node is called the capsule. And this capsule is basically fibrous connective tissue. The lymph node, in addition to this one, is divided into the cortex and medulla. So this outer portion over here, this one is cortex. And this inner portion over here, that's the medulla of the lymph node. So everything that enters first enters the cortex and then it moves to the medulla, and then from medulla it goes out. So you see a lot of afferent lymphatic vessels enter. So those are all afferent lymphatic vessels, and then efferent lymphatic vessels exit the lymph node. It's divided into compartment that's called nodules or lobules, and each lobule is basically packed with dense mass of lymphocytes and macrophages. So if you look at this picture over here, you see a lot of basically lymphocytes, T cells, B cells, and macrophages. So it is that you don't have lymph node as the nervous system, central nervous system. You don't have the lymph node. Cluster of lymph nodes could be found superficially in a number of different regions. For example, right over here. So that's going to be your axillary region. It could be found in the cervical region, right over here. It could be found in the groin region, right over here. So you have superficially and then internally around the gastrointestinal, you have those lymph nodes clustered in addition to being spread all throughout the body. So what is the function? The function of lymphatic node is to basically filter the lymph before it returns to the blood. So it filters it for what? It monitors for any sign of pathogen or infection, so it could basically go and stimulate the B cells and T cells. So B and T cells, B cells could proliferate and produce antibodies. Peyers patch. Peyers patches are basically a round or oval, and they're located around the mucosal membrane of the intestine. So if you look at those ones, so those are all Peyers patches around the small intestine, and you're going to find them all throughout. They're basically isolated lymphoid follicles of gut-associated lymphoid tissue. Their main job is basically to monitor the intestinal lumen and transport luminal antigen and bacteria and stimulate the immune system. So because the lumen of gastrointestinal tract is exposed to the external environment. So if you look at it, it's a tube that runs through our body. So this is basically external environment, the tube runs. So before anything goes in, right over here you have Peyers patch, and they will monitor if there is any pathogen or antigen. They will just inform the other lymphocytes, B cells and T cells, and stimulate them to produce a response. Now, they contain cells that are called M cells, and if you look at this one over here, this picture, so this is basically the lumen of intestine. That's where you have all the digested material, all the enzyme, all the waste. That this waste is going to go through the tube. So if you look at this as your intestine. So this is going to just go out and become waste product. And anything that's beneficial will be absorbed through the intestine and enter the circulation. Now, throughout the intestine, so those are the epithelial cells that you have, but every now and again you have M cells. And those M cells, as if you're holding your hand outside, they will just monitor for antigens. If they do see any antigens, they will go and inform the other lymphocytes and initiate a response. So the way you could look at it, Peyers patch, as if somebody is extending their hand outside and looking at pathogens and sensing the pathogens. And that's, we're going to look at that picture in a second, and something like this. So imagine that this part over here is inside. So you see this guy is standing inside the store and he has his hand outside, and let's see that this was a pathogen right over here. So the guy senses this pathogen, and when it senses this pathogen, this guy will stimulate other, let's say, security people that, hey, we're in danger. That's exactly what happens. So you have the bacteria and everything in the lumen of intestine, and those M cells extend their hand and they sense those or monitor those bacteria. If they feel any pathogenic bacteria, they will go and inform the B cells and T cells, stimulate the T helper cells, and initiate the immune response. So that's how the M cells work. Now, mucosal associated lymphoid tissue are basically the type of tissue that's found all throughout the mucosal membrane in the surface. And those include the Peyers patches, the bronchial, tracheal associated lymphoid tissue, nasal associated lymphoid tissue, vulvogenital associated lymphoid tissue, larynx associated lymphoid tissue. So basically those are lymphoid tissue that's located throughout the body, and their function is that they basically play a major role in regulating the mucosal immunity. So they basically regulating if there is like any sign of pathogen in the mucus, so they could initiate a lymphoid response. And when we say mucosal surface, we know that mucosal membrane is exposed to the external environment. Tonsils are oval-shaped pod that are cluster of lymphatic tissue, and just under the mucosal membrane, they line up the nose, mouth, and throat. So you see some lymphoid tissue all throughout. So right over here, so you see all this different lymphoid tissue. That's the tonsils. They act as a frontline defense, forming the initial immunological response with the upper respiratory system. So when antigens are inhaled or ingested, tonsils are appropriately positioning for exposure. They're appropriately positioned for exposure, which means that as soon as you ingest or inhale pathogenic, that will lead to development or initiation of immune response. They're predominantly composed of B cells, and they have M cells, and you know what is the function of the M cells. They're basically like a hand extending outside and checking the outside environment. They capture basically antigens and activate the B and T cells. And once the T cells are activated, T helper cell will activate B cells, and when the B cells are stimulated, B cells will proliferate, become more numerous, and once they proliferate and become more numerous, some will become plasma B cells and some will become memory B cells. So the plasma B cells are the one that will start producing antibodies. Let's look at the composition of the blood. We already know this. We went through this one, but I'm going to very briefly explain this to you. So basically formed elements, one of the components of formed element is white blood cells. The other two, red blood cells and platelets. We're not going to discuss that. So white blood cells could be either granular or agranular, And granular includes the three type of cells, which is the neutrophils, basophils, eosinophils, and the agranular includes two type of cells. That's the lymphocytes and monocytes. We know that the lymphocytes further divide into three different types of cells. They become B cells, natural killer cells, and T cells, and then B cells differentiate into memory cells and plasma B cells. T cells differentiate into helper cells, suppressor cells, and cytotoxic cells. So let's look at the neutrophils. Neutrophils, which is the most numerous, it's about 70% of the white blood cells, are the very first cells, cell type to travel to the site of infection. They fight infection by ingesting microorganisms, so they're phagocytic cells, and releasing enzymes that kill microorganisms. They also have surface receptors that allow them to detect chemicals such as interleukins, and basically that will allow them to move toward the direction of the infection. Basophils, they're least common. Basophils appear in many specific kind of inflammatory reactions, particularly those caused by allergic symptoms such as asthma. They release anticoagulants such as heparin, and which prevents the blood clotting. They may also release vasodilators such as histamines that will initiate inflammatory response and also vasodilate the blood vessels. Eosinophils, they're basically specialized type of cells within the immune system that are involved in anti-parasitic and inflammatory response. Although they're phagocytic, but they are less efficient in killing intracellular bacteria compared to neutrophils and other cells. Plasma B cells are responsible basically for production of antibodies. Remember, we said B cells differentiate into plasma and memory. B cells will only differentiate into plasma and memory after they have been exposed to an infection and stimulated by T helper cell. So they proliferate and then they differentiate. Natural killer cells kill the pathogen that we're going to look in a second by releasing chemicals. T helper cell basically is responsible for activating the T cells and B cells. So whenever you have infection, it's the T cells that will basically go and say like, hey guys, we're infected. Come on, let's do something about it. The suppressor cells, on the other hand, are the type of cells that will suppress. They go after the infection has been completed. So when your body defeats the infection, the suppressor cells will say, stop. It will deactivate. It will suppress the T cells and B cells and tell them, stop, the infection is gone. Cytotoxic T cells basically destroy virus-infected cells and tumor cells by if the blood vessels, they will migrate to tissue, and once they migrate to tissue, we call them phagocytes. And now what you have is that plasma B cells produce antibodies, and there are 5 types of antibodies: immunoglobulin G, A, M, E, and D that we're going to discuss in a second. And we said those two cells, T helper and T suppressor cell, together, not one of them, but together, they regulate the immune response. So this brings us to our next question. Our next question is specific and non-specific defense. So what is specific and non-specific defense? Everything that you see over here so this is specific defense. Anything outside this is non-specific defense. So neutrophils, basophils, eosinophils, natural killer cells, macrophages, those are all our non-specific defense. And this one, T and B cells are our specific defense. More specifically, it's the antibodies that are very specific and specific defense. So let's look at what is specific and what is non-specific defense. Specific defense will be produced against one specific type of antigen and defend your body specifically about that one pathogen. Non-specific doesn't distinguish between one or other type. They will defend the body against everything. There are 7 different types of non-specific defense. For example, physical fence, physical barrier. Like your skin, your mucosal membrane. So your skin basically serves as a physical fence, physical wall. Doesn't allow bacteria to enter your body. Your skin is not going to be like, hey, if you're E. coli, I will allow you to pass, but if you're staphylococci, I will not. Your skin basically is intact. As long as your skin is intact, it will not allow any bacteria to pass. So it's non-specific. It doesn't say one type should enter, one should not. It will say any foreign object is not allowed. Phagocytes is non-specific defense. So macrophages basically monitor body fluid. So they constantly go and monitor the body fluid and the tissues and check the cells, and they're basically looking for any sign of infection or abnormality, any cell that's abnormal, any protein surface that's abnormal, any cell that basically has a bacterial or viral peptide. Macrophage will go and engulf them. So let's say that the macrophage encounters a bacteria. So macrophage encounters bacteria, macrophage is going to go after this bacteria and engulf this bacteria. So the macrophage is now, you see, engulfing this bacteria. So the bacteria is going to be engulfed by the macrophage. Once the bacteria is inside the macrophage will break this bacteria into small pieces. It will chop it into small pieces. And then we'll take one of those small pieces, one of the bacterial or viral piece, and it will present it. It will go and tell the T cells, T helper cell, that you see this piece of bacteria, this is the type of bacteria that infected us. T helper cell will look at this evidence and go and stimulate the B cell. And it will also present the evidence to the B cell. Now, B cell will produce specific type of antibodies that will only recognize this antigen. If in this course you've been infected by another type of antigen, let's say that the other antigen has this shape antigen, another bacteria. Now, because your antibody were produced against this antigen, those antibodies are useless against this one. So the whole process has to repeat itself. So what does macrophages do? Macrophages constantly monitor the tissue and the body fluid. If they see any sign of infection or infected cell or bacteria or damaged cell, they will go engulf it and destroy it. Natural killer cells, that's the immunological surveillance. That's the third type of non-specific defense. So natural killer cells basically go around and look for cells. So let's say that this is infected cell and this is the natural killer cell. When it says infected cell, it will go move close to the infected cell, and as it moves close to the infected cell, what you're going to see is that the Golgi apparatus over here, this is the Golgi apparatus. Golgi apparatus is going to move toward the infected cell. So you see the Golgi apparatus is rotating and moving toward the infected cell. As it moves toward the infected cell, what is the Golgi apparatus going to do? Golgi apparatus is going to release chemical that's called perforin. So what is perforin is going to do? Perforin is going to go and bind to the membrane of this infected cell and puncture the membrane, plasma membrane of the infected cell, and this infected cell will die. So what is this showing you? So this one over here, let me change the color of the pen. This is the vesicle produced by the Golgi apparatus. And this tiny round molecules are the perforin. So perforin is those round molecules you see inside the vesicle. So this is the perforin. This is the membrane of the natural killer cell. This is the membrane of infected cell. This one is over here, the cytoplasm of infected cell. and this one is the cytoplasm of the natural killer cells. So this molecule is produced, and this is basically the gap between the two cells. So the molecule is produced, this molecule travels, binds to the membrane of infected cell, and makes a puncture, makes a hole in the membrane of infected cell, and the infected cell dies. The next non-specific defense is interference. So interference or cytokines are diverse group of non-antibody proteins. They act as a mediator between cells. In other words, they act as a chemical messenger that coordinate defense of the body. Interference basically bind to the surface of abnormal cell and trigger production of antiviral proteins that prevents proliferation of the virus. The other non-specific defenses complement system. It's a basically group of antimicrobial protein that act together in a cascade fashion to enhance or complement the ability of antibody and phagocytic cells to clear the infection, the damaged cell, or the infected cell. The other type of non-specific is inflammation. Inflammation by itself is a defense mechanism. So what it does is it basically produces a localized tissue response to injury. It basically slows down the spread of pathogen. It temporarily repairs the injury and so forth. So that's all basically, those are some of the symptoms of inflammation. So the blood flow to the region increases, phagocytes becomes activated, capillaries become more permeable, the permeability increases, complement activation occurs, clotting reaction begins to kick in, regional temperature increases, specific defense activation begins to start. Fever is the other or the last of the non-specific defense. Like every other form of inflammation, fever enhances the innate immune defense by stimulating leukocytes to kill pathogens. And also increase in body temperature may also inhibit the growth of many pathogens because now you're disturbing the optimal temperature of the pathogen growth. So let's say, for instance, like you have a pathogen that divides every 10 minutes at 37 degrees of Celsius or 96 degrees of Celsius. Now your body temperature moves to 38 degrees, or let's say to 100 degrees of Fahrenheit or 38 degrees of Celsius. Now that bacteria cannot divide every 10 minutes. It takes 20 minutes. So you slow down the growth of the spread of bacteria. So specific defense, on the other hand, acts on a specific antigen only. And two types of cells are responsible for it. We have the T cells, which basically produces, is responsible for cell-mediated immunity and a defense against abnormal cells and pathogens by attacking them directly. And then you have B cells that provide antibody-mediated immunity, also called humoral immunity, defense against the antigens and pathogens by producing antibodies that will go and bind to the specific antigens and initiate defense system by attracting macrophages. So there are 5 different types of antibodies we have: immunoglobulin A, immunoglobulin G, M, D, and E. Immunoglobulin A is found mostly in secretions like saliva, tears, milk, and so forth. So any of that secretion contains a large quantities of immunoglobulin A. So because it's found in breast milk, that's the kind of immunoglobulin that moms pass to the kids. Immunoglobulin G is the most common type of antibody. It protects against bacterial and viral infection. Immunoglobulin M is found mainly in blood and lymph fluid. It's the very first antibody that will arrive to the site of infection during the first encounter to that infection. And this is the titer of this immunoglobulin will rise first. Immunoglobulin D, one of its functions is to signal and activate B cells. May also play a role in allergic reaction. Immunoglobulin E, which normally is found in a small amount in the blood, they may be higher amount. There may be a higher amount of immunoglobulin E when body overreacts or allergic reaction occurs. So their role is with the allergies. Forms of immunity. What are the different types of forms of immunity? You have two types of immunity. You have either acquired immunity. Acquired immunity is basically after you're born, as you encounter these different situations, you acquire it. You get this immunity. Innate immunity is the immunity that you're born with. So either you're genetically different from some species, prior to birth you were exposed to something and produce antibodies and so forth. So this is the antibodies or immunity that you're born with. Acquired is the one that you get it. Acquired could be either active or it could be passive. Active means that your body actively fights and produces some kind of immunity, fights the infection, fights the pathogen and produces it. Passive means that the body doesn't fight it. It was given to you. So passively you receive that. So what are the two types? Both active and passive could be either induced or natural. So what is natural acquired immunity? Natural acquired immunity, active immunity, you develop after basically you're exposed to some kind of disease. So you become ill with the disease, you actively fight it. So you're ill with the disease, you actively fight that, and then you acquire that immunity against that disease. What would be induced? Induced is, for instance, like example is going to be vaccination. You're vaccinated against some kind of disease. Even after vaccination, you develop slight fever, your body fights it, but then your body develops immunity against that disease. Passive, on the other hand, could also be induced and natural. What is natural passive? When the mom passes immunity to the kids. So mom breastfeed through the breast milk, passes the immunity to the kid. The kid didn't fight it. It was passively given. Consequently, the kid has immunity against that disease. Induced, that would be, say for instance, like if you have been bitten by snake or scorpion or spider, you go to the hospital and the hospital injects you with anti-venom. Those antibodies that you were injected, your body didn't make it. It was passively given and you were induced to it. Properties of immunity. So immunity should be, those are the properties, it should be very specific. It should be versatile, it should have memory, and it should have tolerance. Specific means like it's activated by a response to a specific antigen. Versatility is that it should be ready to confront any antigen at any time. It should remember that antigen, should have some kind of memory. If the same antigen comes the second time, it should be able to recognize and attack it. And tolerance, it should not go crazy and attack your own cells. It should recognize your cells as self. T cell activation. So T cell recognize antigen glycoproteins. Those are complex proteins on the surface of the cell. So we went in our previous lecture that any protein located on the surface of the cell, we call that an antigen. So antigen is basically an ID or recognition. That's how the cells recognize each other, by looking at this one. So if you have T cell over here and this is another infected cell, that T cell will look at this protein and will say, hey, I see something abnormal because this protein is attached to something that I don't recognize. So this is kind of like a recognition or ID. there are two types of MHC or histocompatibility complex. So you have the MHC class protein. There are two classes of this, class 1 and class 2. We're going to start with class 1. Class 1 is found on the surface of all nucleated cells. So all cells in your body that has nucleus has MHC class 1 protein. So this is the MHC class 1 protein. If you remember how the proteins are produced, so you have some kind of DNA inside the nucleus. DNA transcribes into messenger RNA, and then messenger RNA comes out, goes to the RAF endoplasmic reticulum. RAF endoplasmic reticulum translate that messenger RNA into a protein. That protein from the RAF endoplasmic reticulum goes to the endoplasmic reticulum, Golgi apparatus, and Golgi apparatus will pack that protein in a vesicle and transport that. So this is a vesicle, and this structure that you see over here, that's the MHC protein. So that MHC protein goes to the surface of the cell. So this cell, all the nucleated cell, will have some kind of ID card, and that ID card or that identification is this MHC class 1 protein. Let's say that this is macrophages. So the macrophages, let's say that this is the eye of the macrophage. So the macrophage is going to look at this protein, and let's say that this is some kind of cell, whatever cell, muscle cell, cardiac cell, whatever cell. So the macrophages look at the cell, and the message is, you're good. We're not infected. You're good. I'm going to leave you. I'm going to go and check other cells. So that's going to be the message. Now, what if the cell is infected? Some kind of bacteria or pathogen infects the cell. So let's say that this is viral or bacterial DNA inside the cell. So what is going to happen? Consequently, you're going to have a lot of like viral peptides. So you have viral and bacterial peptide inside the cell. Those viral and bacterial peptide will enter or end up in the RAF endoplasmic reticulum. Inside the RAF endoplasmic reticulum, what they're going to do is those MHC class 1 protein will pick up a bacterial peptide. So this blue one that you see is the MHC class 1 protein. This red one that you see is bacterial or viral peptide. So it picks up this viral peptide. That MHC class protein with the viral peptide will travel into the Golgi apparatus. What does the Golgi apparatus do? Golgi apparatus will pack that MHC class protein with the bacterial or viral peptide, and it's going to end up on the surface. So what you see over here is right over here on the surface, MHC class 1 protein with some kind of bacterial or viral peptide. So now what is going to happen? If macrophages or some kind of T cells or other monitoring cells check the infected cell, so this is the infected cell, they will say like, good, I recognize this protein. This is MHC class 1 protein. I'm not going to touch you, but I don't recognize this guy. That's a foreign substance, so that must be an antigen. So in that case, if it is macrophages, what is going to do? Macrophage will engulf the cell and destroy it. So that's how basically MHC class 1 protein work. MHC class 2 proteins, on the other hand, the way they work, the same exact way. So what you have is, you have some kind of nucleus. The nucleus is, inside the nucleus there is DNA. DNA codes for MHC class 2 protein. So they produce messenger RNA. Messenger RNA goes to the RAF endoplasmic reticulum. They're translated to three-dimensional protein. Three-dimensional protein will go to the Golgi apparatus, and then with the vesicle go to the surface. So that's the normal procedure. Now, what if the macrophage encounters or sees a bacteria? So right over here, this is the bacteria. So the macrophage initially doesn't have any bacteria. Now the macrophage encountered this bacteria. So the first thing is going to go surround and engulf this bacteria. So it will phagocyte this infected or cell or bacterial cell or pathogen or any other cell. Once inside, this bacterial cell will end up inside the macrophage. Once inside the macrophage, an organelle called lysosome. Lysosome is going to move close to this engulfed bacteria, release digestive enzymes, and the digestive enzymes going to take and chop this bacteria into small pieces. It will just destroy it. So now what you have is bits and pieces of bacterial cell, bits and pieces of bacterial peptide. At this stage, remember, MHC class 2 proteins are produced. So what is going to happen? This MHC class 2 protein is going to pick up a bacterial or viral peptide and take it and present it to the surface. So you see now it's picking up. So the blue one is MHC class protein and the red one is bacterial peptide. It picked it up and now it's moving to the membrane. So now what happens is the MHC class 2 is presenting the cell. So basically, what is the message here? The message is that MHC class 2 protein will go and tell the T helper cell that, hey, T helper cell, we're infected. There was a bacteria, I destroyed that bacteria. And this is how the protein or peptide or antigen of that bacteria looks like. Go tell the B cells to make antibodies that will recognize this antigen. That's exactly the message. So macrophage is telling the T cell, T helper cell, I found an infection, I found a pathogen, I destroyed it. Here how it looks like. This is the antigen. Here is the antigen. Go tell the B cells to produce antibodies against this antigen. So this is basically T helper cell. So when you look at the T helper cell, this is the macrophage over here. So the T helper cell will look at the macrophage, and in this case I will say like, good news, we're not infected, nothing happens. In this case, what is going to happen? T helper cell will say like, hey, bad news, I see that a bacterial peptide or viral peptide, an antigen, a protein that I don't recognize is attached to MHC class 2 protein. So let me inform other cells. So what is the T cells going to do? T cells will go and activate, T helper cell is going to go and activate T cells and also activate B cells. B cells will start division. B cells will proliferate. And some of those B cells will become plasma B cells and some will become memory B cells. The plasma B cells will start produce antibodies. What are antibodies? So antibodies are basically consist of two parts. Antibodies consist of this part. That's the part that binds to the antigen. And this part of the stimulates the macrophages. So antibodies consist of two parts. This part that stimulates binds to the antigen, two hands, and this part that basically stimulates the macrophages. And what is the function of antibodies? They basically neutralize the pathogen. They activate the complement system. They activate the macrophages and they stimulate the inflammation and prevent basically spread of infection. Now, let's assume that those are the different types of antigens. Each antibody recognizes a specific type of antigen. In this case, if this antibody, let's say that this is bacteria, the antigen of this bacteria looks like this. And this is, let's say, causes pneumonia, this bacteria. So you produce antibodies, so these antibodies will go and bind to this antigen. Now, let's assume that during the course of fighting pneumonia, you encounter another infection. And that infection has, or that bacteria has this type of antigen. So this antibody would be useless against this antigen. For this antigen, you would have to produce another antibody that will recognize it specifically. So what is that telling you? That each antibody is specifically for one antigen. So you see this one, the antibodies recognize specific antigen and basically bind to epitope of that antigen and neutralize the protein. With this, we conclude our lecture.